Ross, Chief Bioscientist at the Franklin Institute. Welcome back to another conversation about the science behind the COVID-19 pandemic. I wanted to touch on a few things that have hit the headlines this week before diving into a more detailed discussion about the emerging antibody testing that's being used to assess how many people in our population have already been affected by the COVID-19 disease. But before we get to that, I wanted to hit two headlines that have kind of emerged above the noise of all of the information that's coming out about the disease. The first one, and I don't think anybody will be surprised by this one, is the science of disinfectants. Now, I'm not the first one to tell you that disinfectants are for killing the virus on surfaces, and under no circumstances should be either ingested or injected into the human body. But there's important things to know about using disinfectants correctly when you are applying them to surfaces. And so I wanted to just touch on a little bit of that to make sure that we're all using these products correctly. So one aspect that not many people might be familiar with is that the EPA has a very specific list of products that have been tested to kill the coronavirus on surfaces. And so you can go and find this list on the internet. It's called List N, N as in now. And this EPA List N gives you not only the products that are effective at killing the virus, but also the contact time that's necessary for this chemical to be applied to a certain surface before it can kill the virus. And that contact time is really important because it's not just enough to kill the virus itself. After a minute or two, there are some compounds that need to be left on a surface for up to 10 minutes before it actually destroys the virus. So obviously that's really important to know to make sure that what you're using is doing the job. Now, whether it's alcohol, whether it's bleach, or whether another common category of chemical disinfectants is called Quaternary ammonium compounds. You might see this listed on a product as alkyl dimethyl ammonium chlorides. It's a lot of big chemical words, but they ultimately they're all derived from ammonia. Now, all of these kill the virus in ways that kind of break up the virus, either that coating that surrounds the virus or the DNA and the proteins that are inside the virus. So one thing that I wanted to touch on as well is another type of disinfectant, which is ultraviolet light. Now, a lot of people have been talking about the ultraviolet light coming from the sun and what role that might play in killing the virus. Now, the problem is that a lot of ultraviolet light does kill the virus, but it also damages our cells. So they're not safe to use in any place where those ultraviolet radiation might come into contact with the human body. As you know, ultraviolet A and B rays come through the atmosphere, and that's why we all have to wear sunscreen to protect our skin from that harmful radiation. There's another category called ultraviolet C radiation that in our environment mostly gets blocked by the ozone layer. So generally we don't have to worry about being exposed to it as much day to day. But scientists are experimenting with how UVC radiation could be used to kill the virus. So there are some places that have already started developing light sources that generate that UVC radiation that could be used, for instance, to disinfect like a subway station at night when there's no one in it. Something like that. But what's really interesting and what's starting to get a little bit more attention is a subcategory of ultraviolet radiation called the Far Uvc radiation. And these are very, very short wavelengths of radiation. So scientists think, at least preliminary experiments in cells outside the body suggest that these types of radiation can't actually damage cells in the body, but could still kill small viruses like the SARS-CoV-2 virus. So we're kind of got our eyes on this one as a potential alternative that could be used in environments like large public spaces to kill the virus with less damage expected to the human body. So that's a disinfectant that is still under experimentation, but something to just keep your eyes out for. Okay, disinfectants, number one. The second set of headlines that I wanted to just touch on briefly was headlines about the emerging drug remdesivir. Now, we've talked about remdesivir on some of our conversations before. It's a drug that already existed as a potential drug to kill other viruses. And so there's been a lot of hope that it might work on the SARS-CoV-2 virus as well to treat COVID-19. Now, the results so far have been mixed. We're just starting to see some results come out of clinical trials, but what we saw last week was some leaked results that came out of China where the clinical trial had to actually be shut down because they didn't have enough patients to get to the numbers that they were looking for. Now, with that, it seemed like there wasn't a significant difference with the remdesivir treatment, but with not enough numbers of patients to compare, and we don't necessarily have the full context of how the research was carried out, we just don't know really how to make sense of that. So I want to caution and still maintain some cautious optimism about the future of remdesivir, because there are some other early studies, both in animals, for example in macaques, which is kind of monkey, as well as a trial in Chicago that wasn't a controlled trial. So again, we still don't have the perfect study to tell us the answer about what remdesivir might be doing. But I think at this point, we need to be cautious about it. It's not dead in the water yet, but we still don't know whether it is effective enough to really ramp up applications of the drug. Okay, so let's pause. I think there's a question.
Yes. So, is it safe to buy ultraviolet lights for personal use? Would that make it better and/or easier than constantly washing our clothes or masks?
Right, that's a good question. In general, I would not advise buying ultraviolet lights for personal use. Depending on the light source, they may or may not filter out the wavelengths that could be harmful to you. As if anybody's ever bought a black light, for instance, that there are lots of warnings about how to apply it. So in general, for personal use, the advice is still wash your hands. Wash them frequently and wash them for 20 seconds, and use those sanitizing chemicals according to the manufacturer's instructions around the house. Okay, so let's move on to this bigger question as we're starting to see some results of antibody testing in different populations. Okay, before I jump into that, I think we've got another question, so let's hold that thought for a minute.
Do you think everyone should have an antibody test? There are people who have this virus and don't even know it.
Oh, okay, so that's a great question about who should get an antibody test. And I'm going to answer that as we start talking about this, so just hold on to that question in your mind. Let me explain really quickly about the rationale behind antibody tests, and we'll get to the answer of who should get it. So again, this is something that we've touched on before, but antibody tests are really starting to ramp up now. We've seen a number of studies start to emerge where entire communities are trying to have antibody testing, and the goal here is to understand how many people in a community have been infected. Now, at this point, because we just don't have the volume of tests to test everybody, what scientists are doing are trying to test random samples, ideally, of a population to get an estimate of who's been tested. So coming back to that question, in an ideal world, if we had tests for everybody, should we be testing everybody? Yeah, that would be great if we could know for sure whether a person has had the infection or not, because as we know, many people who have been infected don't even show symptoms. And so we think that there are many, many more people who have been infected than what our current diagnostic tests show. But we're not there yet. So we'll kind of come back to that idea again in a little bit. But what scientists are trying to do right now is to test a subset of people in a community, and based on how many people test positive and negative in that subset, we can make an estimate of how many people in that whole community might be positive. Let's pause for a question.
So do not ingest a disinfectant or put UVA in your body, right?
Yes, both good takeaway messages here. Don't drink or inject or otherwise consume any kind of chemical disinfectant, and don't use ultraviolet sources of light for disinfection for personal use. I can't stress that enough. So it's a good point to make one more time. Okay, now when we think about these antibody tests that are being applied to a community, to a whole community, ideally what scientists want to know is first, how many people have been infected, because that gives us a sense of, what kind of public health measures we can take. But in addition, it also gives us a better estimate of what the fatality rate is. Now, we've talked about this before, that when we try to understand, how many people die from the disease, it's hard to know the percentage unless you know how many people in total have been infected in the first place. So those are kind of two main reasons why scientists want to get this estimate of how many people have been infected in the community, both to understand the impact of the disease as well as to plan for public health efforts. Now, the problem that we're seeing right now is that the antibody tests that are being used in these studies and that are out on the market are not as accurate as we would like. So a group of scientists that have published their results publicly at a website called COVIDtestingproject.org have been looking at comparing different antibody tests, and they call it, like the head-to-head bake-off of tests, to judge which ones are the most accurate. And when you hear people talking about accuracy, you'll hear people talk about two particular characteristics. One is a sensitivity of a test. And what that means is, for people who are truly positive for the disease, how frequently does the test say they're positive? So this is the true positive rate. So ideally for 100% sensitivity, if you have, for every single person who is definitely has a disease, every 100% of the time, that test will confirm that they have the disease. If there are tests where a person is definitely positive, but the test says it's negative, that starts to lower the sensitivity of the test. Okay, now the flip side of accuracy is called specificity. So how specific is the test? And what this means is how accurately does the test reflect the true negative rate? So for all the people who definitely don't have the disease, ideally the test will 100% of the time turn out negative. Now, when you start having what we call false positives, that a person does not have the disease but the test says they do, that's problematic. Especially in the world that we're living with right now, where we don't have a good sense of who has a disease and who doesn't have the disease just by symptoms alone. So if we end up having a test that says somebody has a disease when they haven't had the disease, then we think that they might be immune, they might be protected, when in fact they're not. And so having a high rate of false positives, so low specificity, is a really big problem. And so what we're seeing with the tests based on the results of the COVID testing project work is that only 3 of the tests that they tested had sensitivity of over 90%. So again, this means that 90% of the time, a person who really is positive is testing positive. And some of the tests had a false positive rate between 11 to 16%. So again, that means 11 to 16% of the people who were testing positive never actually got infected. And when you're dealing with a small percentage of people who have been infected so far, even these what seem like maybe minor problems in specificity and sensitivity can actually lead to really big problems in how we interpret the results. Okay, let's pause for a question.
How soon after you may have had the virus are the antibodies always present?
That's a terrific question. So in this kind of antibody test, how much time does it take for that positive test to show up? So let's remind ourselves how these tests work. So when you're infected with the disease, your immune system starts to generate antibodies, which are cells that recognize the virus very specifically and remember that you've been infected with that virus so that you can respond to it again. Now, it takes some time for this immune response to ramp up. And so what we have seen, again, based on some of the results of the COVID testing project, is that the longer it's been since you got infected, the more accurate the test is likely to be. So certainly, if it's been 2 weeks or so since you first got infected, you're going to get a more accurate testing result than if it was earlier. Another question.
So how do scientists make a vaccine?
Okay, so I'm actually going to pause on that question because we're going to come back to that about how you make a vaccine and how that kind of fits into this landscape of being able to assess who's infected and what that means. So before we get to that question about vaccines and how we develop them, I want to hit on one other challenge that's come up in terms of understanding the results of these antibody tests. Remember how I said that without enough testing or capacity to test everybody, what scientists do is to try and test a sample of the community that they want to understand to get an estimate. Now, ideally, you want to get a random sample of the population that reflects the demographics of the community. So in terms of where people live or what their income status is, men versus women. So all of these, you want your sample to be reflective of the community that you're trying to estimate. Now, that's hard. And scientists, unfortunately, have to take shortcuts. Otherwise, the delay in getting the right sample might push off the useful information that we could use now. Unfortunately, what we've seen is that in some of these studies, people have tried to take some shortcuts that unfortunately compromise the results of the study. So in last week's conversation, we had just learned about a study that was done by some scientists at Stanford University focusing on a community in Santa Clara, California. And unfortunately, part of their recruitment was flawed because it was specifically targeted to a community in a middle school in Santa Clara. And so you're then trying to have to correct for those biases in your sample that then lead to uncertainty in the data. And so, we're still trying to figure out what the best strategy for designing some of these samples are, because if even the recruitment and the population that's included, if that's skewed, then we have a hard time understanding what that means for the whole community. So, what can we tell? Well, one thing is that no matter even with these potentially flawed results, even the highest numbers of infected people in a given community, for instance, some of the studies in New York City, are only at like 20 to 30% at most. So that still means that 70 to 80% of people, even in our hardest hit communities, have not yet been infected. So we've got a long way to go before we start thinking about what we call herd immunity, or how many people in a particular community are protected against the virus, so that that'll help keep infections coming down. Now, getting to the question of a vaccine, the other open question is that we still don't know whether having antibodies means that you are immune against a second infection. And so this is a huge question, not only as we look at people who have recovered from being infected once, but also in terms of getting a vaccine. So a vaccine is essentially simulating what happens when you get infected by a virus. So you can, scientists are trying to develop either a mimic of the virus, or trying to take parts of the virus's genetic code and injecting that into the body so that the body thinks it's been infected and again mounts that immune response. So essentially we're kind of artificially creating an infection that is just less likely to get blown out into a full-bore infection. So whether we're thinking about people who have recovered, whether we're thinking about somebody who's been vaccinated, ultimately we're trying to build up that immune memory in your body so that it'll protect you if you ever get exposed again. Let's pause for a question.
A lot of us are panicking. What is the general timeline for something like this? I think it's causing a lot of anxiety for people. Is there any reassurance?
Yeah, it's an uncertain time. And the reality is that vaccines are hard to create. There's lots of steps along the way at which things may or may not work. So the most optimistic timelines that we're seeing for developing a vaccine is 12 to 18 months. Now, that still seems like a long time. And one of the ideas, and this is why this is a really great question to then segue into the other piece that I wanted to talk about, was how can we speed this up? Knowing that we need a vaccine, as soon as we can get it. So one possibility that scientists have suggested is something called a challenge trial. So let me talk about what that means. So when you have antibodies against a disease, either because you've recovered from it or because you've had a potential vaccine, because some of these early vaccines are already being injected in clinical trials, how do we know that that actually protects you against the disease? Well, one way is to study a huge number of people and hope that some percentage of them get exposed to the disease again, and we can see what happens to their immune response. Now, some people are suggesting that there's a faster way to do it, and this is where challenge trials come in. A challenge trial is when you intentionally infect somebody with a disease. So you would ask for volunteers who want to participate in the challenge trial. Maybe they've recovered already, maybe they've been injected with an experimental vaccine, and in order to understand what happens when they get infected with the virus again, you actively inject them with the virus. You don't just wait around and hope that some fraction of your population gets exposed again. You intentionally inject them so you have a set of participants that you can actually follow. As you can imagine, this is incredibly controversial because intentionally injecting somebody with a disease sounds like it's ethically a little bit questionable. And so there's a lot of discussion right now about when and how this type of challenge trial might be appropriate. Now, I should say that challenge trials, this idea is not new. There are other diseases for which challenge trials have been put into place specifically to test this kind of specific response because it's a very targeted way to do it. And last week, there were 35 members of the House of Representatives wrote a letter to the FDA in support of conducting challenge trials, recognizing the anxiety that's out there in terms of how we move forward, and knowing that a vaccine and understanding whether our immune systems are giving us that long-term immunity, how important that is to move forward. So one advocate of moving forward with a challenge trial, actually right here in Philadelphia, Dr. Stanley Plotkin at the University of Pennsylvania was one of the inventors of the rubella vaccine, and some of the conditions that he's proposed for a challenge trial is to specifically work with young adults, because they tend to have the least severe symptoms to the disease, and maybe we start with a weakened version of the virus. Or just try the spike protein that sticks out of the surface of the virus as a way to start challenging people's immune systems without necessarily exposing them to the full virus. And the question is, do our standards of risk change in the context of a crisis like this? So some people have made analogies that we ask young people to volunteer to fight in wars, knowing that there's a risk of death. Is this something similar? Now, those who argue against starting with challenge trials, some of the arguments against it include the fact that we just don't know enough about the disease right now to know what kind of risk we're asking people to put themselves at stake for. And that, especially with public volunteers, without that detailed level of information, asking them to make an informed decision is really hard. Maybe there's a middle ground there that we start challenge trials with healthcare workers who have a better understanding of the risk that they're putting themselves into and are already at risk, and so that differential of a challenge trial isn't quite as big. So let's pause for a question before I close up with some final thoughts.
Is the Swedish response to this issue being used to refine models? It's 7 weeks and 200,000 have not died. Yes, more have died than, say, Denmark, but Swedish schools, restaurants are open. Doesn't this natural experiment show the initial model assumptions on R factors, CFR, etc. are not right?
So there are two pieces of that question that we can talk about. So as we're seeing different countries' response to different public health measures, we can kind of compare between countries to see what type of impact these different public health measures have on the population's health. And so Sweden has taken a bit of a different approach than some of its neighboring countries. I don't know the numbers off the top of my head, but it does seem that the number of infections and deaths in Sweden does seem to be higher than some of its neighbors in Norway and Denmark. And so when you start thinking about these population level approaches, there are risk-benefit decisions that policymakers have to take. And I'm not an expert in weighing in on what the right decision is, so I don't want to speculate on that front. But in terms of what that means for models, what we know is that models aren't necessarily right or wrong, but models do need to be updated. And so how you incorporate different types of information into that model is something that we do need to keep on top of. That the assumptions that we put into a model 2 weeks ago should not be the same assumptions that we're putting into a model now. And so absolutely, we do need to look at all of the different approaches that different countries are taking and thinking about what that means for updating our models, but also thinking about what are the costs that we're willing to pay. And different societies, different countries may make different choices about that. And I think that kind of leads to this bigger idea that we keep coming back to over and over again, is that when we're talking about pandemics, science by itself doesn't have all the answers. It's a question about taking the science and deciding how to apply it in decisions that are very, very complex, that take into account economics and ethics and just the systems about how our supply chains work and things like that. We're all learning about what systems thinking means as a whole, because all of these different pieces are interconnected. And so, circling back to challenge trials, what does it mean to move forward with challenge trials? What decisions are we going to have to make? And so, again, how can we accelerate the rate of scientific discovery to get us to the end of this, or closer to the end of this? Should we be considering challenge trials in the mix? Sure. What can we do now to get ready for it, so that when the decision is made that yes, we should do it, that we're ready to go? And so there are scientists out there. There's an organization called One Day Sooner, which is an advocacy group that has already started recruiting volunteers with the idea that if challenge trials are approved, then they'll have pre-screened a willing set of volunteers that are ready to go. So, we're inventing all these kinds of new systems and new ways of coordinating science to really speed it up. And I think that's what relieves a little bit of my anxiety, is that we're faced with a lot of unknowns. But the fact that we've seen so much collaboration and so much innovation really start to tackle how we look at this problem in new ways, nothing's guaranteed, of course, but the more solutions that we try, the more likely we are to find something that works. Thanks again for tuning in. We're going to wrap up our conversation about COVID-19 here today. But I want to invite you to join me for a bit more of a fun conversation tomorrow. So tomorrow at 5:30, I'll be joining my colleague Adam Piazza, who's one of our adult education specialists, for a more lighthearted conversation that we're calling the Search Bar, where we'll be talking about science and all sorts of other stuff over a cocktail. So grab your own drink, join us right here on Facebook Live at 5:30. And we'll explore a little bit further afield about where my interests have come from and some of the other scientific topics that are on our minds. So I hope to see you here tomorrow again at 5:30, part of our Franklin at Home programming to keep you engaged as we're all stuck at home. Thanks again.